Hi, my name is Hallie Wiles, and this is my presentation, How Electrodes Read the Human Body. I do give permission for this presentation to be shown publicly. In a single year in the US alone, approximately 647,000 people die from heart disease. What we'd hoped to accomplish with this project was create some kind of early warning device. And what we mean by this is create a device that produces an ECG signal, and not only produces the ECG signal, but is watching and observing the signal, looking for signs of heart defect, signs of heart disease, or signs of a heart attack, and warns the person having the ECG done or wearing the device that they are experiencing these heart problems. And so the first step with our project was learning how the electrodes read the electrical signals from the heart. So this meant doing research into physiology, research into how to read an ECG, and from there we moved forward with producing our own ECG. We were able to produce somewhat of our own ECG. We were able to get a heart signal and be able to see the heart signal itself, but we weren't able to move much further forward than that. Before moving further forward with the presentation, I do want to do a brief heart physiology lesson. The heart is made up of thousands and thousands of cells called myocardial cells. The main job of these myocardial cells is to contract. These contractions happen when ions, specifically calcium, potassium, and sodium, move in and out of the cell. The movement of these ions in and out of the cell creates an electrical difference between the inside and outside of the cell. And so from these differences, we have depolarization and repolarization of the cell. The depolarization and repolarization is what eventually causes the contraction of the cell. Altogether, you're going to have a signal that starts at the SA node in the top right atria of the heart. This signal is going to come from the autonomic nervous system, and it's going to signal the SA node to fire. And the SA node is going to create a signal cascade that's going to tell all of the cells of the heart to contract. It's going to start from the top of the heart and travel down to the bottom of the heart where the ventricles are. And so in the image at left, you can see the arrow shows this pathway of the signal from the top to the bottom of the heart. This signal traveling down is why we see our atria contract first and our ventricles contract second. At right, we see an ECG wave. It includes a couple descriptions of the different parts of the wave. And just kind of looking at it briefly, we can see that there are different heights of peaks. And this makes sense because the atria only needs to pump blood to the ventricles, whereas the ventricles need to pump blood against gravity up through the aorta to the rest of the heart, or to the rest of the body. And so to create this extra large contraction, you need a lot of cells, you need a lot of force. And so that's why we see that increased QRS wave, or that higher peak QRS wave. That is the ventricles contracting. Whereas the P wave, which is the depolarization of the atria, is much smaller because, as I said, you only need to get the blood from the atria to the ventricles. So the rest of the ECG wave is pretty self-explanatory. One thing that I did want to talk about a little bit was when someone has a heart defect, a heart disease, or experiences a heart attack, this ECG wave is going to change. For example, let's say that someone's ventricle has some sort of defect, and so it isn't pumping as hard, or say that there are cells in the ventricles that have died due to a heart attack or something along those lines. This QRS wave that we see, or this peak, is going to be much smaller because if the cells are dead, they can't contract. And so that overall summation of electrical potential is going to be much smaller. And so this was something that we wanted to take advantage of when we were looking at the ECG wave and kind of our ultimate end goal, that our device could see the changes in these ECG waves and alert the person wearing it that there's something wrong. To do this project, we took a couple different approaches. We initially started off thinking that there was something in the electrode pads that would read the signal and produce the signal that we see in the ECG. Turns out, after dismantling a couple different types of these electrodes, there's nothing to them. It's just the cotton, the metal piece that connects to alligator clips, and maybe a piece of plastic disc between the two parts of the middle. So absolutely nothing that could tell us how the signal is read. And so we decided to move forward, and we went and tried to just read the signal with the oscilloscope. And so we did pads on the arm and one on the leg just to ground, and we tried to read the heartbeat just by itself in the oscilloscope. That's all we did. And there was too much noise, we couldn't see anything, and the heartbeat was simply too small of a signal. And so what we did next is we did a bit of Googling, and we found a bunch of different websites that had a lot of helpful information. I believe we Googled homemade ECG, which came up with a lot of different circuits that we kind of gained inspiration from. We eventually found a lab from Michigan State University, seen it right, called Bioelectric Measurements. And this was a very simple, very easy to follow lab where their students were using electrode pads, and they connected them to the upper arms of their patient and one to the leg as ground. And from these electrode pads, there was a connection to a protoboard where they had a differential amplifier, and then that differential amplifier went to the oscilloscope. So in the lab itself, it didn't talk about how to do the differential amplifier, but it was enough to get us thinking and get us on the right track to something a little bit more that would be easier to see on the oscilloscope. As I mentioned in the previous slide, the Michigan State University lab was really good for inspiration on how to get us started with the project. What was not included was how to create a differential amplifier. They simply stated differential amplifier and had one handy. We did not have this handy in the collaboratorium. And so we had to do a little bit of research and reference the textbook to figure out how to create a differential amplifier. What we decided to use was an op-amp, and so what we did is we connected electrode pads to the upper bicep of the left and the right arm, and for our VN and VP of the op-amp, we connected each arm, so left to VN and right to VP, and then our V out was connected to the oscilloscope. And what was created and what V out was the difference between the left arm signal and the right arm signal amplified. And so what we see on the oscilloscope is pictured at right, or I guess GIFT at right, and shows a couple seconds repeating itself of the signal that we saw from this circuit. We were really excited with this signal because before it was just the flat line. We wouldn't see anything. The signal was just simply too small and the noise surrounding us was just too loud. And so we felt really good about getting this signal, and this is where we ended winter quarter before the pandemic and before everything kind of went a little crazy. And so we decided to move forward with thinking a little bit more about the noise that we were experiencing. And the noise is why we don't see the typical ECG wave that I had in the previous slides with the P, QRS, T wave all being very distinct. What was happening in this circuit is that the specifics of the wave were being lost in the noise. And this noise is the environmental noise from the power lines, the 50 to 60 hertz frequency. That kind of disrupts our signal. And so at the point that we were at winter quarter, we knew that this noise was happening. They mentioned it in the Michigan State University lab that the noise was happening, but we were just happy to be getting an amplified signal at all and seeing the heartbeat at all. And so we discussed a little bit more in the future improvements, how we were planning on ridding the signal of this noise. But at this point, we just are able to see that there is a heartbeat. You get that signal, that spike that we see in the GIF, and we were really excited about this. With spring quarter being all online due to the pandemic, we ended up experiencing a lot of issues with our project, and we were not able to move much further forward than what we got to winter quarter. What we struggled with was the fact that due to the stay-at-home order, we weren't able to go to the collaboratorium and work with the equipment that we had originally used winter quarter. And Andrew actually had the equipment necessary to continue working with the device. I did not, and I was not able to go to his house due to the stay-at-home order. And so it came down to Andrew having to work with the device and me just having to be discussing different things with him over text or over the Google Doc. And so this ended up proving very challenging. It was really, I wasn't getting as much straightforward answers as I was hoping. And a couple days before the project was due, Andrew dropped the class and was not able to get me any of the information that I needed to move forward with the project. And so, as unfortunate as this is, I turned around and tried to do the best I could and decided to do a little bit more theoretical since I wasn't able to actually work with the device any further. So what I did is I looked at what we were hoping to improve as of winter quarter, and the first thing that we talked about was Arduino. And so I don't understand Arduino very much. Andrew had a little bit better of an understanding, but essentially what he said we were trying to do was use Arduino to create nice graphs of the signal. And so what I think he was hoping to do was get graphs that we could so that we could see the signal for longer periods of time versus on the oscilloscope where you see it for a small period and you can take a screenshot, but then that's all you get. So with Arduino, we could get a longer period of time so we could observe changes in the heartbeat. We could maybe have someone increase their activity level and observe the changes. We can do different kinds of movement and observe these changes. And so I think that was what his plan was with Arduino. After doing some research, I thought maybe there was a potential application, and again, I don't understand Arduino very fully, but thought perhaps this might be a good idea if it was possible, was to take the data from the graphs created from Arduino and download them and upload them into MATLAB. MATLAB is something I understand a little bit more, and I know that there is code that you can implement that could filter the data that we see from the signals, or perhaps do some kind of if-else statement to see if you can do like if the wave reaches this point, then print heart defect, which is a big reach, I know, but something that I thought about a lot, that if it could be possible, this could be a really big breakthrough. So exciting stuff. Again, might be getting a little ahead of myself here, but if possible, it's definitely something to look more into. Other than Arduino, the other thing that I talked about a little bit in the last couple slides was the need for filtering. And so two of the options that we considered while Andrew and I were still discussing the project at the beginning of the quarter was doing perhaps a band reject or a low pass filter to get rid of the 50 to 60 hertz environmental noise. Both of these are good ideas. Obviously, the band reject would be better because you could get rid of that noise and not have to worry about getting rid of too much stuff. These circuits, I believe Andrew said that he made them, but then I never got any of the information that I needed from him, so I wasn't able to see the results of these circuits that he supposedly built. And so I ended up, for the project report, I created a couple different circuits in PHYSIOLOgy, but wasn't able to run them just a lack of time. I had really hoped to have enough time, but due to Andrew letting me know that, or I guess he never actually let me know that he had dropped the class, but after realizing that Andrew had dropped the class, I didn't have enough time to do anything myself other than get the project done, do a little bit more research, and kind of provide the theoreticals that I've provided. And that brings us to the end of my presentation. Another big thank you to Azad for all your help this quarter and for all your help the entire year. And again, I really hope someone picks this up. It's a really cool project, and I think it can really move forward with someone who has another year on it. Thank you.